So we were talking about the structured coalescent in the context of what's known as the island model, where we had all these identical little subpopulations that have symmetric migration between them. We have D of those little subpopulations, and each one is of size little n. And we worked out what the expected time to, so in the context of that, when we sample alleles, those alleles can be in different states. And in the context of that model, there's really sort of just three states we have to worry about, or kind of two. One is that the two alleles are in the same deme, or that the two alleles are in different demes. And the third state we talked about is when those alleles coalesce. And we worked out that the expected time to coalescent for two alleles from one deme is this, and the time to coalesce for two alleles sampled from different demes is that, which is the same as the first result with this additional component. We could say, what if we just sampled two alleles completely at random, which is what this represents? Well, there's a 1 over d chance that we would get the two alleles from the same deme, and a 1 minus, sorry, that should be 1 minus d over d, or 1 minus 1 over d, that we would get them from different demes. And so we could get the average coalescent time, okay? We're not deriving the full distribution of coalescent times for alleles sampled, but you can imagine that distribution, kind of has these two parts, a distribution that comes from alleles sampled from the same deme, and another distribution for alleles sampled from different demes, okay? The last thing I wanted to finish up with this part is relating this to the idea of FST. Who here has heard of FST before? Yeah, so most of you, right? So you know this is a measure of, or it is a measure of population differentiation that Sewell Wright came up with, and it's often written something like this. Where this is, HT is the heterozygosity in the total population. So in other words, if you were to pick two alleles from anywhere in the population, the chance that they would be different from each other. It's the heterozygosity of the total population minus the average heterozygosity within a subpopulation. So the chance that two alleles within a subpopulation would be different, and then standardized by the total heterozygosity. More formally, you can show that this it reflects the amount of variance in allele frequency across subpopulations. It's worth knowing that's also what it represents. But we can also look at this from a coalescent perspective, because remember, the heterozygosity of two, how different two sites will be depends on the coalescent time, right? It's just proportional to the coalescent time. So from the coalescent perspective, it would be, that heterozygosity is just, you can think of it as two alleles sampled from anywhere. It's going to be proportional to the coalescent time times the mutation rate. But actually, every term here, well, will be multiplied by the mutation rate, so the mutation rate cancels out. So then this is two alleles from state 1, that's the two alleles being in the same deme, divided by that. Okay, so we can get this straight from the coalescent. When we do that in the context of the island model that we've done with a specific number of demes, we get this result. So this is just plugging in all these values and doing a little bit of algebra. You can make it look like this. Squared times 1 plus. So that's with some specific number of demes. And then if you take the limit as the number of demes becomes very large, so in the limit, as d goes to infinity, of this FST, we get this result, 1 over 1 plus 4 mn. Okay. And then, so this is the classic island model result for differentiation. It used to be the case, I don't know if people still do this, but what people would often like to do. So think about what this is. This is the migration rate times the number of individual, or this is the probability of migration times the number of individuals. This represents the number of migrants per generation. And people like that, right? Because they're like, oh, this is, tells us how connected these things are. So people would go and measure FST. So this was something you could go and measure. And then you could rearrange this and solve for this MN, right? The number of migrants. The problem with that is, well, people like to do that. Remember, this result comes from, we start with a very specific model, right? That we have all these demes, that they're all of the same size, that they have symmetric migration, and that we then take the limit as large demes. Okay, so under all those assumptions, then this works out all nicely, and in principle, you could do that and find the number of migrants per generation. In reality, I don't know if there are any real populations that would meet all these assumptions of the island model. And so there, Mike Whitlock, who a professor at UBC, and he was actually taught in the first version of this course, he always jokes that maybe his most cited paper is what he refers to as his but-see paper, because he has a paper where he goes at length and says, this, I think the paper is called FST does not equal 1 over 1 plus 4 mn, because it doesn't for other kinds of population structures and things. And so you have to be very careful about using it that way. And yet people continue, at least at the time he wrote this paper in the early 90s, people would continue to do this all the time, and then they would write but-see Whitlock and Macaulay. so he accumulated a very large number of citations for his but-see paper. Anyway, so some of you who know about FST, and maybe some of you who have used FST, know about this relationship, and that's where it comes from, and just use some caution. One last thing, again, in looking at this, this should make some intuitive sense, right? That when as m gets large, FST goes down, right? Which means we haven't, made a huge, massive mistake in our math somewhere, because that makes sense, right? That populations are going to be more genetically differentiated. More of the genetic variation will be among populations rather than within populations. Like that's what FST is representing, the fraction of variation that is among populations. So FST gets lower as the migration rate gets higher, or conversely, FST gets higher as the migration rate gets lower. Yeah.
So what does it exactly mean to say d to infinity since n is finite? So shrinking the island size?
That's a fair point. I haven't actually plotted this. I don't know what its behavior of, how quickly does it approach this. Maybe I haven't actually plotted it, so I don't know if d of 10 gets you pretty close to this. And by the time you're at D of 30, you're very, very close to it. Or whether you need D of 3,000, I don't know. But of course, it'd be easy for anybody here to just make that plot. Yeah.
So from this result also, it's true that if n increases,
Yep.
then FST would decrease.
That's right, because it.
Biological reason for that.
Yeah, because of what I pointed out before, right? This is the number of migrants that are moving, right? So M is the migration rate, but then the per generation migration probability, the way we talked about it, and N is that. So you're getting more actual individuals taking genes from one place to another, right? You can also, this is again where it's helpful to try and think about all this in a coalescent perspective, right? So if n gets larger, so if you hold m constant, right, that's what you were saying, just increase n. Okay, let's think about coalescence within a single population. What happens to the average coalescence time within a single population if I make n larger? It gets longer, which means it would take longer for you to coalesce, which means the longer it takes, guess what there's more chance for? Migration, right? Yeah, so in sometimes when I look at these kind of parameters, I think about them as this is about the rate of migration, right? The rate, we're thinking about two alleles. The rate of migration is 2m, right? One could or the other one could. And these processes are often about the rate of one force, migration, relative to our other force. And our other force, so let's do it that way. Rate of migration relative to the rate of coalescence. What happens when I rewrite that? Look familiar? There it is, right? So that's the way I kind of think about it. It's the rate of migration relative to the rate of coalescence. Right. Okay. So what people started to realize was, so right now so far in this class when I've been talking about genetic drift and coalescence, I've been thinking about things that sort of affect the whole genome in the same way. But it turns out genetic drift the whole genome doesn't experience genetic drift equally. And one of the simplest reasons would be, say, comparing the sex chromosomes to the autosomes. And that's just a simple numbers game, right? There's fewer Y chromosomes in a population than there are a given autosome, right? And same, there's fewer mitochondria than there are autosomes. There's fewer X chromosomes than there is for a given copy of an autosome. So just for simple number reasons, we would know that some parts of the genome experience different amounts of drift and thus coalesce at different rates. But at a much finer scale, people started to realize that neutral sites that don't affect selection, would be affected by selection occurring elsewhere in the genome. And in some way, we actually have already seen a result that's very conceptually very closely related to this, which is we derived a result where out came the variance in fitness, right? The outcome, the variance in fitness was a key component in determining how strongly drift was felt. And if you think about a neutral site, it's the amount of variance in fitness it experiences will in part depend on what genetic background it's in, right? So if I imagine, I'm one neutral allele and Christian is another neutral allele, so we have no merit unto ourselves. We're just neutral passengers. And one of us happens to be lucky and be with a good fitness variant, and one of us happens to be unlucky with a bad fitness variant. We experience this variance in fitness because of that. Unlike environmental variance, which you might imagine occurs sort of randomly anew each generation, that linked variation can persist for multiple generations. So there's a covariance in the fitness across generations. And so a site closely linked, a neutral site closely linked to a site under selection experiences that selection for a longer amount of time than a site that's further away and has a greater chance of recombining away from it. So this turns out to be quite important, and I was going to talk about that in detail. I'm instead going to talk about it just qualitatively with respect to negative selection, so selection occurring on deleterious mutations when they arise. And the effect can be even stronger when you talk about beneficial mutations. But I think Guy is going to talk about that next time. But I want to give you a bit of a feel for this background selection result. So here's the way that I sometimes think about it. So imagine, my pointer is not working here. It is not switched on. Turns out there's some major technical challenges for me. Okay, so imagine you sample these two alleles, and then these two, this allele, if we're thinking backwards in time, so this is sort of real time going forwards, this allele, is randomly choosing a parental source to come from. That's, a conceptual way to, and so is this one. And so there's all these possibilities for them to choose among, right? And so that's why the chance that they would choose the same one would be 1 over 2n. But now let's imagine that what we can't see is that there are these bad genetic backgrounds. Okay, so what's shown here are the focal sites, and in gray we're imagining there are these deleterious mutations that occur somewhere nearby, and those so here's one that occurred, and it persists for a while, but because they're deleterious, they don't last that long. So they drop out of the population. So if ignoring for now recombination, let's imagine there's no recombination in this population, then if we think about these alleles going, each generation jumping back and randomly choosing a parent, well, we kind of know, none of these could have come from this part, right? Because this thing is doomed to die out. It's sort of from the moment it starts, sometimes it's been called like the walking dead, right? It's not going to persist into the future very long. Yeah, so in the absence of recombination, all of these backgrounds are inaccessible. And so if you think about that, instead of there being 2n choices, there's a smaller number of choices each generation. And so if there's a smaller number of choices, that of course means that coalescence will occur more quickly. Or thinking in forward in time, it means the amount of drift experienced by these ones is stronger. If you have recombination, then they're not complete, this is not a completely inaccessible background, because it could have been on here and then recombined off. But the way you can think about it is it's kind of a hard to get to background. It's not as easy to get to this background as it is to get to that background, because this requires this additional thing of having to recombine onto it during the time where this lineage even exists. Okay. So, it turns out we can do this in the coalescent framework, and this was worked out in the mid-90s, and I've given you in detail sort of how to do this, but I just want to give you just a little bit of a flavor for it, because it turns out doing this is conceptually very close, in some ways identical, to the structure, to the geographical structure model that we did, the island model. Really, when we have some selected site, which I'll call it the B locus, and then we have our neutral site. Which I'll just represent with a square. Whoops. And so this might be the wild type allele that has some fitness 1, and this is this big B allele could mutate into this little B allele, which has a fitness that's reduced by some amount HS. And so when we think about our neutral allele, it can either be in this kind of a background, right, a genetic background, or in this kind of a genetic background. And so that sort of naturally sets up something very much like having our physical patches, right? We can have, if we sample two of our two samples of our neutral allele, those samples could be in a few different states. They could both be on a big B background, on the good genetic background. So that would be what I might call state 1. They could both be found on a bad genetic background. Or one could be found on a good genetic background, and the other one could be found on a bad genetic background. Okay? So conceptually, right, just like what we had done with the other ones, we identified what states our samples could be in. Before we had a total of 2 states, and here we would have essentially a total of 4 states if you include the coalesced state. And so we can approach this problem just like we approached the other problem. And again, we have to work out those transition probabilities of changing from one state to the other. And so I'm not going to go through this in detail, but it's all in the notes if you're interested. In the physical model, one of the key things of how we moved from things being in the same deme to being in different demes was because of migration, right? Now what we do is the way you move from being in one kind of background to being in another kind of background is first via recombination. Right? You can have a recombination event. So if your sample was sitting there, but that individual was in this current generation in a heterozygote, then it would have the opportunity, or in the previous generation was in a heterozygote, it would have a chance to recombine onto a new background. So that's one way. And the other way is via mutation. Okay? So you have to account for the selection that's occurring, which the selection against the little b allele, which plays a major role in determining how frequent this bad genetic background is in the population. Right? The frequency of this bad mutation is going to depend on the rate of mutation creating it and also the selection against it eliminating it. okay? But you can go through and you can work out these transition probabilities. And this, if you do all this, which is given in the notes, you can work out what the average time to coalescence is. And you get this result. 1 plus. You get this reasonably famous result, which says this is the normal coalescent time, right? 2n, the expected coalescent time for two alleles. And then it's multiplied by this part, which is 1 minus this little bit, right? So if this little bit wasn't there, we'd have the classic result. And so it's this little bit where Q is the frequency of that deleterious allele at mutation selection balance. So that's that we didn't talk about this in class, but at mutation selection balance, you can show, at least in a deterministic model, that the allele frequency of the deleterious allele is the mutation rate divided by the selection coefficient. So it depends on the frequency of the deleterious variant, the recombination rate between your focal site and your deleterious site, and the strength of selection against that. It turns out if you look just at so this is the effect of a single site, a single selected site on the coalescence of a neutral site. But of course, there's probably many selected sites near a neutral site, right? So if you think about a neutral synonymous site sitting in a gene, there's probably going to be lots of selected sites around it, and they're all contributing this kind of a thing. And so you can, if you're willing to make a few assumptions, you can integrate across a region and ask what's the total effect on the expected coalescent time of our neutral site. And you end up getting this. Where big U represents the total rate of deleterious mutation over the stretch that you're looking at. M represents the length of the stretch that you're looking at in Morgans, so in recombination units. So this is kind of like the density of deleterious mutations occurring in a region, okay? So this, I should say that this works in a region where recombination rate maps linearly with distance. And as those of you who know more genetics know that if you look over longer distances, that doesn't work because of crossover interference and other things. Okay, but this is, so what this can mean is in regions, so if you think, so let's think about two kinds of regions. Regions that are gene rich and regions that are gene poor. So if you're a neutral site sitting in a gene poor region, then there's not going to be many selected sites around you. If you're sitting, so this density of deleterious mutation, so the rate of deleterious mutations per map length will be low. You aren't going to have to worry too much about this kind of thing. But if you're in a gene-rich region, then this is potentially a much larger effect. So even if you were looking at purely neutral sites across the genome, you would expect different levels of standing variation, right? Because the neutral sites sitting in gene-rich regions experience more background selection, which you can think of as a lower effective population size. Right? So they will tend to coalesce more quickly and thus be able to retain at the population level less genetic variation than another neutral site sitting somewhere else in the genome that is not surrounded by the same density of deleterious mutations occurring. One practical concern that people have raised about this is, well, what that when one looks at when one looks at, say, different populations and finds diversity, you find more differentiation at some parts of the genome than other parts of the genome. Our natural inclination most of the time is to go, oh, those must be regions that are selected differently, right? So that they're selected differently, and so they've differentiated more than some other parts of the genome. But this is kind of a very boring alternative explanation, right? That some regions of the genome experience more background selection, and so the coalescence events will occur more quickly for them, right? They will, in other words, you're more likely to get coalescence events within populations than you are alleles from currently within a population are more likely to coalesce within that population rather than to be able to coalesce with a sample from your other population. Whereas regions that are experiencing less background selection are less likely to coalesce. On average, will take longer to coalesce within a population, providing more opportunity for them to migrate out and coalesce with one of your samples from a different population. And so that's what that can lead to different levels of differentiation or different levels of FST for regions that have experienced strong background selection versus weak background selection. Okay. Okay. Any questions about any of that, either qualitatively or quantitatively? Okay. So I'm going to switch gears completely now. If I can. Okay. Let me see how I'm doing for time. Okay. Oops. So I'm not going to do any more coalescence or really any more writing on the board and showing you equations, trying to walk you through those. I realized there was one thing I wanted to say that I forgot to say that came up at lunch the other day that I probably should have said right at the beginning. I always tell my students when I'm teaching them theoretical biology that I sort of think of the process as involving 3 steps, and maybe it will be helpful for you to think this way. And so when you start with some question, and then the first step of that question, the first phase is taking that question and translating it, that biological question, translating it into some starting set of equations. So that's kind of the first process of, how do we do that? So if you go back and think about the things we did, we start with an idea, and then we translate that into a set of equations. The second phase is what I think of as the technical phase, right? You've got some starting set of equations, and now you've got to, sort of solve them in some way or work with them. And that's where it helps being a good mathematician, right? So you do a bunch of math stuff, and so depending on how good of a mathematician you are, sort of depends how much of this kind of stuff you can do. So like Kavita can probably do a lot more of that than I can. She's a much better mathematician than I am. And then the third step is you've got a result. And sometimes people forget about this third step, which is, now that you've got a result, let's look at that result and try and interpret it biologically. What does it mean? Right? So I wish I had sort of laid out these 3 steps in the beginning, so you, at least implicitly, you could have thought about them as we were doing each of these things. But I did try and do all of those steps with you guys. And I always find that helpful to keep that in mind, and maybe you will too, even when you're reading the literature, that sometimes when you read a paper, you try and grasp what's the idea and how is it being translated into a starting set of equations. If the paper is written by some hardcore theoretician, you might not have the technical background to follow all the but then at the end, out comes the result, and then at least try and see, what does this result mean? Does it make sense? How does it relate to the starting assumptions we had right at the beginning? Okay? Okay, so what I want to talk about for my remaining time today and tomorrow is a topic that I have become increasingly interested in the last while. I haven't actually produced very much on this topic myself, but a number of students in my lab are working on this topic now, and I'm interested in it, and so I'm forcing the rest of you guys to learn something about it too. Hopefully you'll find it interesting. So it's about intralocus sexual conflict. So I'm sure many of you know that in many species, males and females obtain fitness in somewhat different ways. And it's of course worth noting that there's lots of organisms that actually don't have separate sexes at all. But of course many animals and some plants do have separate sexes. And so, males and females obtain fitness in different ways. For example, variation in females might primarily come from variation in the ability to obtain food and convert that into offspring, whereas variation in males may primarily be due to variation in the number of females they mate with. Of course, the almost stereotypical example for this kind of idea are peacocks and peahens, and no more appropriate place to show that than in India. But it's native here, right? I want to point out, sort of before I continue, that the standard roles played by each sex differ a lot among species. Just as one example, in North America, you find a lot of frogs in this family, and their females produce large egg masses, and the males compete to mate with as many females as possible, and nobody provides any care. But there's lots of other different kinds of frogs. There's really cool glass frogs. I don't know if you can see this glass frog here. This is a bad photo, but a photo I took in Costa Rica a couple of years ago. That's a male. So in this species, females provide limited form of care, and in many species, or in some species in the family, females provide a limited form of care. In other species like this one, the males provide extensive care. So this is the male, and this is an egg mass that he's guarding. And they do these cool kicks when the wasps come and try and eat the eggs. Anyways, the point is, we might have certain ideas about sex roles, and anyways, nature does not necessarily conform to the stereotypical one that you might have in your head. But the key point for this whole idea is is not what those roles are for males and females, but just that they're different from one another, right? That the phenotype that's best for maximizing male fitness will often not be the same as what it is for maximizing female fitness. And it's also worth saying, although I'm not going to talk about it, that within a single species, there may well be multiple ways to be a successful male or a successful female. But for simplicity, I'm going to ignore that, and we're just going to say there's sort of one way to be the best kind of male and one way to be the best kind of female, and they're different from each other. Okay, so here's really the problem. So selection might favor different phenotypes in males and females, and how do you produce phenotypes using a shared set of genes? So remember, most genes are autosomal. They're not on sex chromosomes. Many traits that could be selected differently between the sexes have the potential to be affected by autosomal genes, right? Guy has just shown you a mountain of data from human GWAS studies that, pick your trait of interest and he can show you genetic factors affecting it across the whole genome, right? They're not, in fact, I think most of the figures he showed you excluded the X chromosome entirely, right? So those were all autosomes. And the other thing is that actually alleles tend to have similar phenotypic effects on both males and on males and females. So in other words, the taller allele, if you're looking at height, tends to increase height regardless of whether if that allele is sitting in a male or sitting in a female. So here's potentially the problem. If males and females are selected in opposite directions for a trait, imagine males are selected to be taller and females to be smaller, but if alleles have similar effects on both sexes, then that, of course, means alleles will have opposite fitness effects in males and females. And so that gives rise to this idea of what is these days called intralocus sexual conflict, or IASC, which means that one allele is favored in males and the alternative is favored in females. So what's going to happen to polymorphism at a site experiencing intralocus sexual conflict? Okay. So for the sake of simplicity, for everything I'm going to talk about here, I'm going to more or less ignore genetic drift and return to our happier world where we just worry about selection. Okay. Okay. So let's start with this first simple case. So imagine we have this A2 allele which increases fitness in males relative to some other allele A1, but has no effect in females, right? So in other words, here's how it affects males. It increases fitness by 10%. So that's 10% would be a massively huge selective advantage. And in females, it has no effect. So what's likely to happen to the A2 allele? It's going to spread to fixation, right? Like ignoring genetic drift, it would spread to fixation. Okay, what about this next case, where the A2 allele still has this same large beneficial effect in males, but has a very small deleterious effect in females? So here it's causing this big advantage in males and this very tiny disadvantage in females. Your intuition will be correct, I think. Pardon me? We'll hang around the population. So here where it was -0, we all agreed, select it will get rid of. So how small would this number have to be for you to give me the same answer you gave up here? So only if it's 0. So if it's 0, we can work out this way. Any value greater than 0, you're going to tell me it's going to hang around the population? Well, I'm saying you start with as a polymorphism and then this selection happens. Yeah, it does. And the equilibrium is it's gone. The equilibrium point is this large advantage in one sex determines is much larger than the disadvantage here. Okay? So it turns out I was wrong because I was like, your intuition will probably be correct. Turns out your intuition is not necessarily correct. So, but that's actually why I do this example, because many people think that if there's conflicting selection of any sort, that you would maintain polymorphism. That's not true. So that's why I do this advantage. I first start with it has a big effect in one and no effect in the other. That case is simple and intuitive. This case, where you have a very small disadvantage, isn't enough to maintain a polymorphism. Okay? So yeah, question.
What if it would have been otherwise in the second case? I mean,
If I just reverse swap this value with this value, it'll be this, I mean, the opposite, you get the same result. It doesn't matter, I mean, the other way of saying it is if I just switch these labels, yeah? Yeah, it's the same result, right? It doesn't matter that it happens to be advantageous in males. If it had a big advantage in females and a small disadvantage in males, same result, same thing. Right? So just because there's some kind of opposing selection does not mean you will maintain variation. I mean, here's another kind of sort of almost analogous example, is if I had an allele that increased female fecundity by a large amount and caused a very small decrease in female survivorship, you would probably all easily agree that that kind of allele would be net beneficial and spread to fixation. The alternative would be wiped out. This is really almost the same thing. It's just now the effect is spread, instead of being trading off between two fitness components within an individual, it's trading off between fitness components when that allele is found in different individuals. Okay? Or another example, which is actually very, very closely related to everything I'll talk about, is imagine you had two kinds of environments, a drier environment and a wetter environment. And if you had an allele that created a large fitness advantage in the drier environment, but came at a very slight cost in the wetter environment, and individuals sort of randomly found themselves in one of those two environments, right? You would probably intuitively guess that, well, if it had a large advantage in one environment and only a small disadvantage in the other environment, and that allele is going to find itself about half the time in one environment, half the time in the other, you're just going to average across the two and go, oh, well, on average, it's a good thing to have, right? So my key point, because I feel like I'm belaboring this, but people often think just because there's any opposition that you would maintain a polymorphism. That isn't true.
I still don't understand why. Like, I trust you, but
Yeah.
Give me some math calculation. Like, why would A2 go extinct?
Well, it's essentially that. So you can think about it just like the physical environment thing, right? That that allele is randomly finding itself half the time in one and half the time in the other. So you just kind of ask what's the average effect of that allele, and that's right. Yeah, I'm sorry. So did I say this backward? The A2 allele is going to spread to fixation.
Oh, yeah, A2 goes to fixation.
Yeah.
That's what it was.
Yeah.
Oh, I see. A2. Yeah.
said it was expanding variation.
Oh, yeah, no, I was wrong. Yeah, A2 goes to fixation.
Yeah. Okay. Okay. So in that example, I had this very extreme difference in what the advantage was in one sex compared to the disadvantage in the other sex. But what if the magnitude of the beneficial effect in one sex is not so different than the deleterious effect in the other? Well, there's really three possibilities. One is that one allele will fix, or the other allele will fix, or selection will actively maintain both alleles in the population. And that's this last possibility is an example of what we call a protected polymorphism, which means that selection is preventing either allele from reaching fixation and thus maintaining the polymorphism in the population. Okay, so here is kind of the setup for that kind of model. We imagine there's some, I've changed the symbols now. I have big A alleles and little a alleles. And I want to emphasize, for evolutionary biologists, a lowercase allele does not necessarily mean it's recessive. Okay, so I know that's the tradition in classical genetics, and so I often find, especially with my undergrads, that as soon as they see a lowercase allele, they assume that means it's recessive. That does not, that's just a label. It doesn't imply that it's recessive. Okay, so this is, we can think of this as kind of the reference allele. We give it a fitness of 1. This allele causes a disadvantage of SM in males. So the M, meaning it's referring to the selection that occurs in males. And we use this HM to control the dominance of that allele, right? Are you guys familiar with, if I remove the M's off here, are you familiar with this kind of fitness model? Put your hand up if you are familiar with this kind of fitness model. Okay. Huh? I'm sorry, I can't hear.
She has a doubt.
You have a doubt. You have a doubt about this. Yes, but this is just an autosome. I'll talk about that in a minute. You're jumping ahead. So this controls the dominance. So if you make this 0, then the big A allele is dominant. If you make it 1, then the little a allele is dominant. And if you make it 1/2, it's exactly intermediate. Or that's what we call the additive model, same additive model that Guy was talking about. Okay, so that's how this allele affects fitness in males. We do the same but opposite thing for how it affects fitness in females. Okay, so you can work through this model. So you were asking how you work through this mathematically. We're doing a model that only involves selection, so you can figure out the frequency of any genotype G. So this F prime means the frequency after selection is equal to the frequency of that genotype before selection times its relative fitness. So relative fitness is its actual fitness value, as given in this table, divided by the mean fitness. And in this context, I'm meaning all sort of sex-specific. So you would be doing this separately for males and for females. And when you do relative fitness, it's relative to the mean fitness in males or the mean fitness in females as appropriate. So one can easily use this kind of model to figure out what the frequencies of the 3 genotypes would be after selection, given some starting allele frequencies. And then you can figure out what the frequency of the big A alleles. If you know what the genotype frequencies are after selection, it's easy to calculate what the allele frequencies are after selection. So this represents all the alleles that are found in the homozygous big A males, and then half the alleles that come from the heterozygotes after selection, and the same kind of thing for females. Right. If you were actually going through analyzing this model, so this model we started really with 2 variables, the frequency of the big A allele in the in the males at birth and in the females at birth. Oh, sorry, this is actually, I said that wrong. This is the frequency in the male gamete pool that created the population, and the frequency of big A in the female gamete pool that created the population. And it turns out, rather than using those 2 variables, that it's better to change variables and use the average allele frequency and the difference in allele frequency. But you can find recursions for how these things change with across generations, just as a function of the recursions I've given you here. Okay, so this is old stuff that people have done a long time ago, though there's been a resurgence of interest in this recently. So you can do this, and you can find equilibrium. So essentially, you just want to solve the system when the values after selection are equal to the values before selection, right? So that you reach an equilibrium, there's no further change being caused. And a simple inspection of when you look at these kinds of equations is that the so-called boundary points are equilibrium. In other words, where one allele is fixed or the other allele is fixed, those are equilibria. And the question, well, let me come back to that for a second. There may well be other kinds of equilibria, polymorphic equilibria. Those can sometimes be very difficult to identify, or particularly to analyze the stability of those equilibrium points. Although they're analytically tractable if you're willing to make some assumptions, like selection is weak or the effects are additive or something. But we can actually just focus on these two equilibria, which are much simpler, and study their stability. And so the idea is, are those boundary points stable or are they unstable? And if they're unstable, that means anytime you get close to the boundary, you're going to be held away from that boundary point. And so if we can, essentially the idea is let's see if we can find conditions where both of the boundary points are unstable, and that will mean that selection is going to protect the polymorphism, or at least it should do in this deterministic model. Okay. So I'm not going to go into how one does this kind of stability analysis, though you can do it this way by just taking the derivative of the equation for change with respect to the variable. And I encourage you, anybody who's really interested in learning how to do theoretical biology, to if you can possibly afford it, buy this Otto and Day book. It's a truly magnificent book if you're interested in learning theoretical biology. Yeah, a really wonderful resource. Okay, so that's basically how you would analyze the model, but let's just look at what happens when you do that. You find that in order for this boundary point to be unstable, you need to satisfy this inequality. In order for the other boundary point to be unstable, you need to satisfy this inequality. Putting those together, both inequalities are satisfied when you get this, when this relationship holds. So let's just look at what that is. So here is so here is selection. The selection that occurs on females, and here is the selection that occurs on males. And this shaded region represents when this condition is satisfied, under the assumption. Here I've made the assumption that this should say HM and HF, but when those are both equal to a half. And what you see is maybe what your, somewhat what your intuition would have told you, right? So you tend to be stable along that diagonal, which represents where selection in one sex is balanced in magnitude by the opposing selection in the other sex. But sort of one bit of foreshadowing here of something I'll get to in a minute is we think that the vast majority of mutations have small effects on fitness. So while I've shown you the whole range from 0 to 1, 0 to 1 on both axes, we think most mutations sort of occur around here. And you can see there's almost no part of a very small fraction of that space satisfies this condition to maintain a polymorphism. However, you can vastly broaden this if you make a different assumption about dominance. And actually, I'm going to move on and I'll return to that later. Okay, so I want to make one note of caution. The model that I've talked about before is kind of a simple, completely deterministic model. If you build in genetic drift into that model, which Tim Connellan and Andy Clark have done a few papers on this, you will find that even though you're supposed to maintain a polymorphism, and in some cases you can work out that there's a stable interior equilibrium, and you're supposed to have an allele frequency at some particular allele frequency, because of genetic drift, you may drift away from that, and in fact, you might reach fixation. So this figure sort of shows a dramatic case of that, where here in a large population, so this is using this kind of a scaled selection coefficient that Guy was mentioning before. And again, you can kind of think of all these things as or I like to think of them as it's selection relative to the force of genetic drift, right? You get something representing selection on the top and 1 over n or 1 over 2n or something on the bottom. And so when you put that together, you get some ns kind of thing. So this is when selection is strong relative to drift. These different color shaded curves represent different scenarios of what the equilibrium frequency is supposed to be. So in this example, the equilibrium frequency is supposed to be 1/2. And even with genetic drift, you usually find this shows the distribution of where you're likely to find yourself. And you stay, somewhere that looks pretty good, right? Pretty close to 1/2. Or in this case, it's supposed to be at 0.75, and it's pretty close to that. But as you make the effective population size smaller, you get less and less of your distribution is at that equilibrium point. And if selection is weak relative to drift, then actually most of the time you are very close to the boundaries and may well have lost the variant completely. Okay, so that's just a kind of a caveat that those deterministic models might tell you that you're maintaining variation, but if you have ignored genetic drift, in reality, that variation won't necessarily, the opposing selective forces might, on their own, might have maintained polymorphism, but they're not enough to maintain a polymorphism in the face of genetic drift as well. Okay. That one-locus theoretical model that we did, the one that led to this result, I think there's sort of 3 really simple but important points that come out of it. It shows that antagonistic alleles will often spread to fixation, right? There was all that white space on those figures, right? Not much of it was made of that shaded region. And so anywhere in that white space represents an antagonistic allele where we expect, even in the deterministic case, to lose variation. And so what that means is that one sex is kind of getting the allele that it wants, in quotes, and the other sex is getting stuck with an unfavorable allele. On the other hand, there are those shaded regions, and so that means sexual antagonism can, under the right conditions, create a protected polymorphism and thereby contribute to the maintenance of genetic variation for fitness in each sex. And of course, this relates very much to one of the fundamental questions in our field is, why do we have genetic variation for fitness or fitness components? Which that question traces back to the very early part of evolutionary biology becoming a scientific field. Regardless of whether one allele has fixed or whether the polymorphism is protected and segregating, it means that one or both sexes are prevented from 100% of the time carrying the allele that is most beneficial for that sex. Okay. So I will here mention something about the genomic location, where we might expect to find the genomic location of sexually antagonistic variations. So we worked through, or we didn't work through, I talked about this autosomal model, but you could also think about genes on the X chromosome. You're not going to have sexual antagonism on the Y chromosome because the Y chromosome never under conflict. It's only ever in males. Well, with rare, that's not entirely true actually, but in the simplest case, that's true. So for an X-linked chromosome, we would have the same fitness model for females as we had here, but for males, we don't have heterozygotes, right? So we just have, males are either big A or little a, because they only carry one copy of the X. And so this is another, so if you make the assumption, of what's called equal dominance or parallel dominance, that I'll return to in a second, then this is the result for maintaining a polymorphism in the autosomal case, and this is it in the X-linked case, and of course those don't really mean anything to you, so I just want to show you what that translates to in a figure. So here's a figure that shows that, and so again, one axis is selection in females, the other axis is the strength of selection in males. And the shaded region shows where you would maintain the polymorphism in the autosomal case. And then it would be inside these different dashed lines that would show where you would maintain a polymorphism if the locus was on the X chromosome, and it's showing you for different levels of dominance. And so that's this panel. The panel on the right is actually just shows the same thing, but it's zoomed in on this weak selection area. Okay, so this goes from 0 to 0.1 and 0 to 0.1 as opposed to 0 to 1. And so here you can really see that in this autosomal case, it's very hard, a very small fraction of this space is amenable to maintaining a polymorphism. But provided that the allele that's deleterious in females is recessive in females, an HF of less than a half, there's a much larger space where you can maintain variation. And so that led to the prediction that you would expect to find much more of this sexually antagonistic variation segregating in natural, or segregating in populations would be located on the X chromosome rather than the autosomes, because it's so much easier for you to fall into the right part of parameter space. I want to point out that this comes from a bit of a weird this is done under this particular assumption of what I refer to as equal dominance. And what it means is that if the big A allele is dominant, that it's dominant in males and it's dominant in females. Conversely, if the little a was the one we thought dominant, that it would be dominant in males and it would be dominant in females in the same equal way. And that seemed a very sensible assumption to people. The early classic work on this topic was this 1977 Kettlewell paper that I've cited here. And this work focusing on autosomes versus sex chromosomes comes from this Bill Rice paper in 1984, and both are based on that assumption of equal dominance between the sexes. And so the idea is when you have equal, that equal dominance only matters for the autosomal case because there is no dominance for the males. Right? And it's intuitively easy to understand why you can maintain variation in the X case if the deleterious allele is recessive in females, right? Because as the allele that's bad for females becomes rare, selection will only ever see it in the heterozygous state in females, right? Or almost, the vast majority of time that selection sees that allele, it will find it in a heterozygote. And if it's recessive or mostly recessive, selection, it has little to no effect on fitness in those females, right? And so as long as the allele that's bad for females is recessive, it can easily hide out. And so you just have to worry about, does it have an advantage to males? I'm speaking a bit loosely here, but that's why this region's so wide, right? If that allele, if it's largely recessive in females, then there's all this extra space. But under this assumption of equal dominance, that won't work for the autosomal case, right? Because if the allele that's bad for females is recessive, then the same allele is recessive in males, but that's the good allele, right? And so then that's a problem, right? Because you get that sort of, you can't say the same, make that same argument that I made earlier, both when a given allele is rare and when it's very common. Okay. So that's kind of this idea of why in this autosomal case, under that assumption, this is so narrow, and the autosomal case is so broad. Okay, so that led to this prediction that we tend to find sexually that the X chromosome would be this enriched for this sexually antagonistic variation. So I want to show you a paper from Adam Chippendale when he was actually in this guy Bill Rice's lab, and he did this really nice experiment in fruit flies. And I'm going to sort of skip the technical details of how they did this, but essentially using some of the tricks that are available in Drosophila melanogaster, they could isolate sort of entire haplotypes. And by entire haplotypes, I mean from a fly population, you can sort of pull out, flies have sort of 3 major chromosomes, one of which is the sex chromosome. You can pull out sort of a copy of the X, a copy of chromosome 2, and a copy of chromosome 3, and that represents one haplotype. And then you can do that again and make another haplotype and another haplotype, and then you can you can make as many copies of that haplotype that you want to. That's my 10-minute warning, I think. So they did that, and they can take those haplotypes, a given haplotype, and then they can make male offspring with that haplotype, and they can make female offspring with that haplotype. So that's what they did with this set of, I can't remember, something like 40 haplotypes. And they measured what juvenile fitness, so I think it's basically egg to adult survivorship was, for these different, so each of these points represents one of these different haplotypes. And so this is how they affected the juvenile fitness of females and the juvenile fitness of males. And clearly there's this strong positive relationship, right? Haplotypes that are good for one sex are also good for the other. Those of you who know much about fruit flies know that the fruit fly larvae are, they, at least at the gross level, they seem to be more or less the same, regardless of whether they're male or female. So it's perhaps not surprising at all that during the phase of their life where they are largely the same and are largely doing the same kinds of things, eating food and growing, that they're affected in the same way. But when they looked at the adult fitness component, they found a strikingly different relationship between how those haplotypes affected male fitness and how they affected female fitness. And this they sort of integrated across those two fitness components, juvenile and adult. And here's the fitness rank of how they affected male fitness and female fitness. And you can see that, this is not, if these were positively correlated, you would expect all these essentially parallel lines. And what you tend to see, what's quite obvious here, is there are a lot of lines that have low fitness in one sex, but those correspond to a high fitness in the other, and vice versa. Okay, so this was quite a dramatic result and piece of evidence suggesting that sexually antagonistic variants must be reasonably common, or at least playing a big role in affecting the variance in genetic variance in fitness. They did a later study where they just isolated the effect of the X chromosome. So I mentioned that flies have 3 major chromosomes, one of which is the X chromosome. And so they did another study to kind of essentially to isolate or to investigate how much of that pattern they'd seen using sort of full haplotypes was driven by the X chromosome. Because remember, 15 years earlier, Bill Rice had written this theory paper predicting that sexually antagonistic variation should primarily be on the X chromosome. Well, what's shown in dark here is the effect of the entire haplotype, the X and the autosomes, and in the light gray, the X chromosome alone. And he put on this dashed line to represent the 20% benchmark, because essentially the X chromosome represents about 20% of the genome of fruit flies. And what you see, so first, this is the additive genetic variance in fitness for females and for males. And you can see the contribution of the whole genome and just the X. But you can see the X is contributing more than just, it only should be contributing about this much based on that 20%, and it's contributing more than that. This is the same kind of data for males. Skip this, but and here's the kind of maybe the key part is the how much of the intersexual genetic covariance. And so how fitness in one sex is related to a gene's effects on the other sex. And there you can see that the contribution from the entire haplotype is actually just equal to what it would be just from the X chromosome alone. In other words, the interpretation here is that entire negative relationship comes just from the X chromosome for adult fitness. Okay, so that's one piece of evidence in support of that kind of variation being enriched on the X chromosome. But there's conflicting evidence of this, and I should say there's a nice review by Jim in this Jim Fry paper of this. So here's a different study which also is evidence of this sexually antagonistic variation. So Fedorka and Mousseau, they went and collected crickets from the field. And they captured males that were either actively mating with females, or ones that weren't. Right? So essentially what they had were males that they knew, based on that observation, were more fit or less fit in the field. Then they took those crickets and mated them to females in the lab and asked about about how the fitness of their offspring. So these were the males that they caught that were mating, the successful males, and these are the males that were unsuccessful, that weren't mating. And what you can see is, if we focus first on the fitness of their sons, is successful males produce sons that are more fit than the sons produced by unsuccessful males. Okay, that maybe makes sense. But you get the opposite result with respect to the daughters, which is what you would expect under this idea that sexually antagonistic variation for fitness is occurring. Right? If you're a good male, that means the idea is that the alleles that make you a better than average male would make you a worse than average female. And so when you produce daughters, they're less fit than these other males. This is quite a striking result. Now, if you think about this, well, what does this mean about X chromosomes? Like, can we say anything about X chromosomes or autosomes? Well, if you think about it, males don't transmit X chromosomes to their sons, right? So if this was driven entirely by sexually antagonistic variation sitting on the X chromosome, then a good male would be good because of those male beneficial alleles sitting on his X chromosome. But guess what? He doesn't give that X chromosome to his sons. So no benefits, right? So the fact that you've got this result for both sons and for daughters, to some extent implies that this crossing pattern strongly implies sexually antagonistic variation. But the fact that you're seeing it for the sons also implies that it's not actually sitting on the X chromosome, there must be a substantial autosomal component. Okay? So maybe something different in crickets than in flies. Okay, so, right. So that's a bit of a puzzle then, right? Because I made this whole point of we can really, here's for more believable selection between 0 and 0.1. And again, even these values are very high. I mean, really, we should probably be looking somewhere in this range or even lower for most mutations. So how is it that we can be maintaining, if we are, and it looks like from those studies that we are maintaining sexually antagonistic variation, how can we do that on the autosomes? Like, you have to fall in that teeny tiny little space. Well, this is where I want to come back to this and remind you that these figures are based on this idea of equal dominance, that the allele that's dominant in one sex, that same allele is dominant in the other sex, which was a reasonable assumption, and people really thought, well, in the absence of anything else, that seems the most reasonable thing to do. But there's some reasons to think that might not be the way things work. So this figure here just points out, again, it's kind of like the very first one I showed you. So in the solid lines is a repeat of essentially the figure I just showed you. So again, this is the full range of selection, and here is focused in on a smaller range of selection. So that solid line, there's that very narrow space where you might maintain a polymorphism under equal dominance or parallel dominance. And the dashed line captures, and the gray part represents all the extra parameter space where you can maintain a polymorphism if instead of having equal dominance, you had what's known as a dominance reversal. It's specifically the allele that is good in males is largely dominant in males, and but the other allele, the allele that's good in females, is largely dominant in females. Or said the other way, the allele that's bad in a sex is partially recessive. To draw these figures, we assume that they had a dominance coefficient of 1/4, right? So remember, 0 would mean completely recessive, 1/2 would mean the heterozygote is exactly intermediate, so 1/4 is sort of halfway in between those two. Okay, so there's all this additional space if you can get if this dominance reversal occurs. Which on the one hand might seem really weird, right? Like, why is it that one allele would be dominant in one sex and the other allele would be dominant in the other sex? Right? Which is why people largely ignored that possibility for many years. So let's talk about dominance. So, and where we're going with this is that maybe dominance reversals evolve. So we can't sort of get into that without noting that Fisher and Wright famously argued about why deleterious alleles tend to be recessive. In fact, as far as I understand it, they corresponded in quite a friendly way up until they started disagreeing about why deleterious alleles tend to be recessive, and they had a big falling out over this issue. So Fisher argued that selection would cause deleterious alleles to evolve to be recessive. And Wright argued essentially that selection would be too weak. Selection only has the opportunity to cause the evolution of dominance when it sees one of these alleles in a heterozygote. But deleterious alleles are held at a low frequency because they're deleterious, right? And so the selection for changing the dominance on those alleles is only going to be on the order of the mutation rate creating those deleterious alleles and keeping them or adding them into the population all the time. So Wright said Fisher couldn't be right about alleles evolving to be about dominance evolving. Instead, Wright just argued it was probably essentially some mechanistic or physiological reason for why deleterious alleles tend to be recessive. And we tend to think that that was largely right. Sort of historically, Brian Charlesworth pointed out that Fisher's argument couldn't explain the observed negative correlation between how dominant allele was and how what its homozygous selective effect was. So what we tend to observe is that alleles that are like lethal alleles tend to be highly recessive, and alleles that have small effects on fitness are partially recessive or almost additive. And that you wouldn't predict that relationship under Fisher's argument. And maybe the most convincing piece of evidence was Orr in '91 did a survey of mutations known from Chlamydomonas, this unicellular organism that lives normally as a haploid. So there should be no selection on dominance of those kinds of things because the thing only exists as haploids. However, under some conditions, it makes diploids, which are dead ends. There's not really any selection on the diploid stage as I understand it. But you can still measure what the effect of mutations are in diploids. And so he surveyed the literature and he found even though there should really be no evolution for effects in diploids, you still find mutations tend to be recessive in this haploid organism. Okay, so this whole argument between Fisher and Wright, or this whole argument between Fisher and Wright, where Wright was saying dominance would evolve, Wright had argued right from the beginning that seemed unlikely because the strength of selection acting to modify the dominance would be so weak because the deleterious alleles are so rare to begin with. I guess 20 years ago now, Saliato and Denis Bourget pointed out that that negligible selection argument wouldn't apply to alleles that were maintained at intermediate frequency, right? Because then selection would be seeing heterozygotes a lot of times, and there would be much more opportunity for the evolution of dominance. And so they did some nice models of that, in sort of building off that, Spencer and Priest 10 years ago specifically examined this case of sexually antagonistic variation that we're talking about. And I had thought I might go through this in detail. It's a bit of a complicated model, but I'll just summarize one point, which is what they found was you could get the evolution of dominance at a sexually antagonistic allele, and you would tend to evolve these dominance reversals, okay? But there's a couple of caveats. One is their model requires that you have a stable polymorphism to begin with, and so it's kind of like, well, the dominance reversal will help you be a stable polymorphism, but in order to evolve a stable polymorphism, you need to be stable to begin with. So that's sort of which came first, the chicken or the egg kind of issue. And the second is that selection on modifiers tends to be very weak, and so in finite populations, even though selection might favor them to evolve, they won't necessarily evolve. Nonetheless, we're now starting to see a few examples. So this is a few examples of dominance reversals. Whoa, I'm way over time again. Okay. We'll talk about that next time. So see you tomorrow.